Hello, today we have 12 questions to be discussed. Now we can discuss the first questions. Why the researchers remove interscapular BAT and not another? For this question, I didn't find a precise reason. I will introduce a bit about BAT and express my opinion. iBAT resembles a primate gland regulating the output of a variety of hormones. BAT function may depend on its anatomical location and development region. iBAT regulates acute macronutrient metabolism, while perivascular BAT regulates vascular function. In iBAT, an upregulation of genes involved in insulin signaling and lipid metabolism was accompanied by enrichment of lipid-related processes and functions, plus glucogen and peroxisome proliferation activated receptor signaling pathways. iBAT constitutes approximately 1/3 of BAT mass in rodents. So my opinion is, firstly, iBAT is big enough, easy to operate, and when remove it, it may have enough functions. The second is maybe some hormones will affect the results. So when remove iBAT, the data
Wait. Zixuan, let's discuss it together, okay? So you're talking about the why the brown adipose tissue from this interscapular region was taken, right? So basically, this is the most common way to use that tissue to use the brown adipose tissue, and like the most easily accessible way to do that. So that is basically the reason. So that's just a model. So they kind of use the iBAT in order to extrapolate that to all others. So just as an example. So, but I think the message is pretty clear. So what they showed. So I mean, you don't need to take all of others, you know. So to do that, you just take the major depot, the major one,
yes,
and then try to see what you see. Yeah, but I think it's a valid question. So it means that the contribution of the interscapular brown adipose tissue is much more valuable than others, which are much smaller, and that means that it is really enough to manipulate the phenotype by just using that one. Yeah.
Yes.
So if anyone has additional questions or comments, you're welcome to say on both sides. Okay.
Okay. The second question is, did the authors mention about the miR-378a by PAR-seq and HIT clip?
Yeah, that was my question. Yeah.
I didn't find it in the database because the reviewer mentioned the literature. So the reviewer said P11O alpha is already a validated target of miR-378a.
The reviewer of this paper.
Yes.
So they have they have shared also the comments and the responses of the reviewers.
Yes, they shared a peer reviewer.
Okay.
The first they just said that it is a possible candidate, and the reviewer said provide this literature and say it's already validated.
Wait, but in the paper they explicitly write, okay, since it is already an established target, so we went there and there. And why you say that the reviewers told that to them? Because
I mean, they firstly think it's a candidate. And they, in their original manuscript, they write it as a candidate gene.
Wait, but usually if you just think it is a candidate, you go make all of these kind of studies to prove that it is a real candidate, to make this dual luciferase assay or hits clip or anything else. And they they submitted the manuscript without having all that, saying that it is just predicted. They didn't do any validation, and they didn't even know that it was validated in other literature. Is it?
Other literature, I forgot to search it.
But so in the paper, in the text of the paper, they refer to that. They say it is already a validated target. And you say that this one was pointed actually by reviewers, so they suggested that. And my question is, did they go for publication in Nature Communications without having?
There might be some literature to prove that, to prove the luciferase experiment result. But I forgot to search it. I just.
Because for me it is very important here, explicitly for this paper, and probably I will tell it later why. Okay, anyway, so the link, this reference where they refer to the previous literature where they already validated this as a target. What was the method used for validation?
You mean this literature?
Yeah.
Luciferase.
Dual luciferase assay.
And just a traditional miRNA study method. And they studied about using Western blot.
Western blot.
Study the mechanism, the pathway.
Okay. Okay. And that was in which tissue? Okay, so they didn't do hits. So hits clip, you say, was not found to be checked. Yeah. Okay, we will talk about that a little bit later. That was, I just explained again. So my question to Zixuan was, how did they validate? Because they say that it was previously already known that P110 alpha is a validated target. So I asked, how did they validate? And Zhang says it's dual luciferase assay, some additional Western, but not hits clip or parsec. Okay, or par clip, how's it called? Okay, okay. Then we can return to that later.
The second question is third question. Do they provide the CT levels of serum miR-378a? They didn't provide the raw CT value, just the CT value normalized with U6. And how are they compared to other serum miRs? And heat map data is from database. Other figures data are from their own experiment. They just compare the miR expression level in room temperature and the cold house mass.
Here I had a concern because the majority of microRNAs in the hepatocytes in the liver are microRNA 128, 22A, 22B. So most of the Argonaute 2 protein within the RISC complex is bound to those microRNAs. So there is just very little left for some other additional microRNAs, especially those which are maybe not even synthesized within the cell, but just came from the bloodstream, right? So from the brown adipose tissue, as in the form of vesicles. So can you imagine? So the liver synthesizes like very huge amounts of microRNA 122, A, B, also some other microRNAs, and then less of others. And then you bring additional amount of some those extracellular vesicles with some even if it is like huge amount in terms of like brown adipose tissue or the serum, it won't be so much that the liver can synthesize itself. So it won't be more than microRNA 122. So that's why I have a question. Like how, if you compare this microRNA expression, so what is the kind of relative expression relative to other microRNAs measured in the liver? Whether they measured, whether at least they show some CT values, because this one, like what they show, like one, the level 1 and the level 1.5, this could be very low. Like the base could be very low, so the functionality is questionable. I really don't understand how does it work functionally. How can this small amount of microRNA work if there is not enough AGO2 available in the liver?
But that's exactly what we discussed for my project, for example. Like that some microRNAs, some monoclonal RNA can work in very low concentrations, can have an effect on very low concentrations.
How? No, just tell me how. I just don't understand how, like what is the mechanics? MicroRNA doesn't work by itself. MicroRNA doesn't inhibit the expression of the mRNA, or doesn't exert, like microRNA is just a guide to make the scanner, which is the risk. Risk is a scanner. It scans different mRNAs for the match with the microRNA, which is loaded to the AGO.
Because maybe that microRNA targets a mRNA that other microRNA don't target. That's the point.
But targeting this, the RNA interference requires AGO-NOT. AGO-NOT is the driver. It is something which makes which stalls the translation, something which degrades the mRNA. MicroRNA doesn't do it by itself.
My point is like maybe there is very few risk complex or argonaut risk complex available.
Yes.
And just with that very bit can work.
And that is my question. So it doesn't seem plausible, feasible. I don't understand the mechanics. So that is my question. That is my major point. Also, that is my question about this Hits clip. Why I asked about this Hits clip. If they could show that in the cold condition, you can see that there is indeed binding of the target with the microRNA 378 within the liver tissue, then I will believe it. I will say, okay, yeah, you basically you cross-link the microRNA with the target mRNA. You then remove this cross-linking. You sequence. You see both the target and this 378, and you clearly see, yes, they target each other. Then I will say, yes, it works. Without that, like everything what they show in vitro, I mean, of course, like you have huge amounts of everything. You artificially overexpress the microRNA, or just do it in vitro, or just add it and so on and so on. So of course you can get all the effects you want. The question is what happens in vivo. So that is a big question for me. Okay, so but we can probably return again to that one when we discuss the overall conclusion of the whole story. Then we can discuss. But if anyone wants to add something to that, so we can discuss that. But by the way, I just want to confirm again. Fazli and everyone on the other side, you can hear Zixuan well, and everyone else here. Okay, that's perfect. That's perfect. That's perfect. Okay, Zixuan, please continue.
Next question is the influence of down-regulated microRNAs. In this paper, researchers didn't explain the down-regulated microRNAs function. Because I think in their opinion, ideal functional BDEV microRNA would be expressed by BAT at moderate or high levels, and increased in fact BDEVs and serum EVs upon cold exposure.
Wait, I don't quite understand the question. So the influence of downregulated microRNAs.
Because there are some microRNAs
Oh,
downregulated.
oh, I see. Uh-huh. Okay. Okay.
Okay, next. Glycogenolysis related genes expression. For example, the glucose output from hepatocytes. Was it only from gluconeogenesis or both states and the glycogenolysis? Authors didn't detect glycogenolysis related genes. For my opinion, is maybe because of the fasting before sacrifice. In a fasted state, the liver secretes glucose through both breakdown of glycogen and de novo glucose synthesis. During prolonged fasting, hepatic gluconeogenesis is the primary source of the endogenous glucose production. So I think the glycogenolysis doesn't happen in their research.
Well, but they do detect one of the genes which is inhibiting glycogen synthesis. Yeah, so the glycogen synthase is being phosphorylated by the glycogen synthase kinase, and that is one of the genes which they found as one of the key ones to to prove that indeed so gluconeogenesis is increased, right? So we're talking about the that's what Zixuan was just saying. She says that they did not address the genes related to glycogenolysis, so that they didn't address or they didn't study.
What's the difference?
I mean, one thing is they checked and that it is not changed. The other thing is that they didn't check.
They didn't check.
They didn't check those genes. Yeah. So that is the answer. Yes, I'm trying to find the had a nice overview. Okay.
Go on.
Yep. Let's go on.
The sixth question is cytokines and their actions on pancreas and insulin production or secretion for target cells. Cytokines are cytokines secreted by brown adipose tissue that can regulate the metabolism and thermogenesis of target cells, such as the liver, muscle, and brain. miRNAs are a type of cytokines that can modulate the expression of genes involved in glucose and lipid homeostasis. Some of the miRNAs that have been identified as cytokines are miR-92A, miR-133B, miR-378, and miR-348. The actions of cytokines, especially miRNAs, on the pancreas and insulin production or secretion for target cells are not well understood. But here are some studies suggesting the possible mechanisms. The miR-92A was shown to be secreted by BAT and to target the insulin receptor substrate 1 gene in the liver, thereby reducing insulin sensitivity and increasing glucose production. And miR-133B was found to be released by BAT and to enhance insulin sensitivity and glucose uptake in skeletal muscle by targeting the suppressor of cytokine signaling 3 gene. And miR-378 was reported to be expressed in BAT and to stimulate insulin secretion from pancreatic beta cells by targeting P10. miR-348 was suggested to be involved in BAT thermogenesis and to inhibit insulin secretion from pancreatic beta cells by targeting the gluconeogenic peptide 1 receptor gene. Those findings indicate that cytokines, especially miRNAs, can have diverse and complex effects on the pancreas and insulin production or secretion for bucket cells, depending on their target genes and tissues. However, more research is needed to elucidate the exact mechanisms and pathways of cytokine signaling and regulation. It's okay?
Nice overview, thank you. I'm just looking at the miRNA 378, which was reported to target PTEN. And PTEN is doing exactly the opposite as the PI3 kinase. So they are working on the same reaction. So on the PI3 either phosphorylation or dephosphorylation of so they are catalyzing the opposite reactions. So this means that the same miRNA is targeting both the direct and the opposite and the reverse reaction. So I think we can hear you very well. Yeah. Okay. So I just asked the question about the miRNA 378 because it seems to target both the direct and the opposite regulator of the PI3 kinase pathway, so to phosphorylate the PI3. So phosphonidyl 3-phosphate, like from the phosphonidyl diphosphate. So the question is, how's with that? So if the same microRNA actually works on both, like how does it? How does the system discriminate? Like in which case it goes towards the increase, and in which case it goes towards the decrease? So that is.
But this happens. Yeah, it happened, for example, also with miRNA 29. It target NRAS and also target PTEN. So probably the discrimination will be on the type of binding, no? Like the seed itself, if it has copied the molecule.
I think the point here is about the kinetics and about the grade, how strongly it does interact with one or with another. Yeah. It depends on this seed region, like how well does it bind, also how well does it guide the cleavage, and so on and so on. So my point is, like, this needs to be addressed. We need to find out, like, what is the rate limiting here, like does it first target PTEN upon increasing concentration, or does it first target PI3 kinase, or there is some kind of context-dependent mechanisms? So this is kind of unclear. So this raises some questions to fully understand the whole system. Okay. That's a great question. I mean, and we are here talking about like cell autonomous or even endocrine action here, right? So like cell autonomously, there could be some types or subtypes of effects, or even paracrine effects. But in this case, we are talking even about like very distant interaction. Yeah. So the brown adipose tissue working on the liver, right? So, and it is. It's a great question. So it's very interesting phenomenon what they are describing here. But in order to make bold claims, I think you need to have very, very, very strong arguments, very, very solid facts to support that. Absolutely. But for me, the key is that we need to demonstrate that the mechanism which they describe is really working, and it is it is what they claim it is mediating this effect and not something else. But we can maybe come to that later. You have also your paper there, your PDF, so that we can later zoom in into the screen. Yeah, okay, that's great. Okay. Yeah. Okay, Fazli, it's a great question, but I think we need to address those really one by one and test all of these hypotheses really one by one to check which exactly microRNA working on which exactly part of the insulin secretion in which exactly condition and to limit all other factors, the influence of all other factors, like what is related to cold, what is related to physical activity, and I think only then we can have a clear picture. Thank you for the nice comment. Yeah, I think I think these authors here convincingly show that indeed the temperature does influence the expression, if not just expression, maybe packaging, or maybe both, expression and packaging of the microRNAs to these extracellular vesicles. I think this had been shown very convincingly. So I am convinced, at least, so that in the brown adipose tissue, indeed, we have the upregulation of the secretion of this miR-3 378, and this is amazing. This is really nice. I really like how they prove it, and I don't have questions regarding that part. I rather have questions about how those microRNAs work on the liver, and I'm not so convinced, but maybe we can come to that a little bit later. Okay, Zixuan, should we continue? Yeah.
Now is the introduction about the EV secretion inhibitor GW4869. GW4869 is a chemical inhibitor of axonal biogenesis and secretion that works by blocking the activity of neutral sphingomyelinase 2, NSMase2, an enzyme that catalyzes the hydrolysis of sphingomyelin to ceramide. Ceramide is a lipid molecule that is involved in the formation of multivesicles bodies, which are intracellular organelles containing exons. By inhibiting NSMase2, GW4869 reduces the levels of ceramide and disrupts the MVB formation, thereby preventing the generation and release of exons from various cell types. GW4869 does not affect the packaging of specific molecules into exons, but rather inhibits the overall production of exons. And next is can we use the BDEV system to package proteins or microRNAs of interest? EVs are lipid membranous vesicles. It participates in diverse biological processes such as cell differentiation, proliferation. EVs can effectively deliver cargo, including drugs and nucleic acids, to targeted tissues or organs because of their transportation capability and ability to cross biological barriers, such as the blood-brain barrier. EVs can be used in the drug delivery system to carry bio-drugs or chemical drugs into the pathological tissues, as they possess several natural advantages, such as the natural barriers traversing capacity, intrinsic cell targeting properties, and the stability in the circulation. And after these BDEVs are excreted into the bloodstream, how are they reabsorbed by which organs? Due to the lipid membrane structure, EV can be reabsorbed by endocytosis. Until now, there is no conclusion about how does EV target cells. Some studies prove that the EV can be non-selectively incorporated into various types of recipient cells. However, another study showed that there is a possibility that cellular targeting is based on signaling molecules. such as morphogen, with a source sink mechanism being suggested. Indeed, a biodistribution study shows that red blood cell-derived EVs predominantly target the liver and bone. Also, melanoma-derived EVs were mainly taken up by the lungs and spleen. As well as organ targeting, EVs have a natural targeting capability based on donor cells. Also, that's why the researchers labeled the BDEVA to track it in this paper. So back to the initial question, there are still some challenges when using BDEV. Difficult to control its distributions, targeting, or delivery. Let me go to the.
I think the data about how these vesicles end up in the liver, and also this miRNA ends up in the liver, is very convincing. Also for me, looks like what these authors of the paper showed. Really nice labeling of this palmitoylated GFP. Really beautiful experiments to withdraw the brown adipose tissue and to see like how does it change. So this is really nice, and it is kind of even a little bit surprising. Like this you know, that you have the whole bloodstream, you have secretion of multiple vesicles from different tissues probably, right? And you have like, they are filled with some different miRNAs, and then you get those ones which are specifically from the brown adipose tissue containing this 378 can manage to reach liver. And the question is it really, like, since liver is known to absorb like quite many different substances, so, and also is used for elimination of many drugs and many biological metabolites. Is it because like liver is so metabolically active that it can absorb basically everything what is in the bloodstream, so it will end up in the liver, or it is just that that they didn't check other organs, and maybe the other organs can also get those signals? So that is an interesting question, which they didn't address.
They check other organs.
Check organs for what? For
You mean where the BDEVs arrive?
Yeah.
Many organs, but not very. The amount is low.
You mean in the liver is the amount is more, the amount which is uptaken by the liver, right?
Yes.
Okay, that is what I assumed, yeah, because liver is so metabolically active, so to basically scavenge everything what is in the blood, whatever, like all these metabolites. So probably that's supplement, right? Supplementary?
Supplementary.
Yeah. Okay, that's interesting. So that's supplementary figure 3, right?
Yes.
Uh-huh. It's difficult to see. Uh-huh. So this is also this palmitoylated GFP, right? So this the same method that they use.
Yes.
Heart, kidney, inguinal fat, right? I assume. Interesting. Really interesting. Uh-huh.
It seems there is quite a lot in heart, no?
Yeah. Not bad. Yes. So it's and we don't know what and that is only the vesicles which are uptaken from the brown adipose tissue, because they did the experiment like that. You can imagine like maybe some other organs also secrete whatever. So this might be even more complex, and they contain, of course, different types of carriers. not carriers, but like the microRNA or maybe even proteins, or who knows. Okay, okay. Interesting. Within the EVs, the microRNAs are transported without ago.
Without ago.
Question it. The question.
Didn't.
Because they mention it. You mean they didn't check? They didn't check.
In their literature, I didn't find information about it.
Because that could be the explanation. So if it is transported already as a complex within the ego, then I could understand how could it work on the liver. And so for me, this is the key question. So without this, I kind of, for me, it is hard to really understand the mechanics. How does it work in the liver with such a small number of microRNA? Because what they show is they increase, like upon cold exposure, the brown adipose tissue enhances the expression maybe 2.5 times, like the exocytosis of those 378. So we detect also increase of the plasma, also around 2 times, only 2 times. But this means that if the base is very low, 2 times is nothing. I mean, this is, if you have very low, you have 2 times very low, so it is still very low. Same in the liver. You have mostly 122A, 122B, some other microRNAs in the liver. All other microRNAs are much in a lower abundance. And there it comes. There is 378, which is 2 times higher than very low. So it is still very low. How can this very low concentration of microRNA 378 in the liver have a physiological function if the ego is basically all taken up by these microRNAs which are highly expressed? So that is a really question, like I don't see the explanation. They don't provide the explanation, and it seems that the reviewers of this paper did not look into that, or so they just believe that the story as it is. So that is my major question, actually.
Yeah, it makes sense that it was transported with the ago.
With the ago, yes, that could be an explanation, but they don't show this, according to Zixuan. Right? So my question is, within the EVs, is this microRNA transported with or without ago? In the complex of with ago or without?
I don't know. I didn't focus on pay attention to it.
Because I think they mention it in a paper somewhere in the beginning. They mentioned this, but I didn't see that they provide any data which are ruling one or another. So that is my major question for this paper, actually. Okay, but maybe we can look into that. But meanwhile, maybe you can continue, right?
Yes. And last question is, how do you think the BAT can selectively package one microRNA over another one? The selective packaging of microRNAs into BDEV is complicated and poorly understood process that involve multiple factors. I think such as the microRNA sequence structure, expression level, and its interaction with the RNA binding proteins can affect the selectively package. Also, it can be regulated by some genes. For examples, HNRNPA2B1 was shown to be involved in the sorting of long RNA transcription to EVs. However, the exact mechanism and the rules of microRNA selection and packaging into BDEV are still unclear and require further investigation. Yeah,
there are really nice studies about how the loop of the microRNA can dictate whether these microRNA remain more stable or less stable in neurons, and also like the same is hypothesized also for the packaging into the vesicles. So it is really interesting to address, but in this paper they didn't focus on that.
Can I see the previous slide? I just wanna.
Next question is the comment on browning of VAT fat as a tumor treatment. Browning of VAT fat as a tumor treatment is a novel and promising strategy that aims to exploit the metabolic and thermogenic properties of brown fat to starve and shrink cancer cells. Brown fat can burn glucose and fatty acids to produce heat, which can reduce the availability of these nutrients for tumor growth. Moreover, brown fat can secrete various factors such as cytokines and macrophages that can modulate the immune system and the tumor microenvironment. However, this approach has some challenges and limitations. For example, the low amount and the activity of brown fat in adult humans, especially in obese or elderly individuals. The next question is, what about hot weather or conditions and the metabolism, gluconeogenesis? If this research is translated to humans, many of them live in regions with hot weather. Also, your thoughts on global warming and metabolic adaptation in evolutionary context. I found some literature prove that hot weather or conditions can affect the metabolism of organisms. And especially the species depend on the external temperature to regulate their body temperature. One of the metabolic pathways that can be influenced by heat is gluconeogenesis. But I think this research result is far away from translated to humans. For example, this paper used the male mice to study. It didn't study about the female mice because in female mice the hormone changes a lot. Also, they studied in 25. The temperature they studied is 25 degrees, and the cold temperature is 4 degrees, which is very mild. But in Pakistan, the 40 degree is really hot. I think they use 25 degree and 4 degree to study just didn't change too much for the mass for the mass. But if they increase the temperature, some results will be changed. That's my opinion. And for the evolutionary context about the metabolic adaptation of global warming, I think the easiest opinion is that we will evolutionary to have some abilities to make us alive from the. That's the explanation.
That's interesting observation. I like interesting thought about the why they use only males. I suppose since females like having higher estradiol levels, which is acting to suppress the AMPK, which in turn acts to suppress the sympathetic nervous system, which in turn is working to increase the thermogenesis pathway. So it might be that in their case, so this effect might be somehow distorted and enhanced because the females, they intrinsically have like more disinhibited effect. So this AMPK influence on inhibition of this sympathetic and thermogenesis pathway is blunted because of this estradiol. Because in the ventromedial hypothalamic nucleus, estradiol inhibits AMPK pathway. So that maybe that is why. So they didn't want to overcomplicate this by having also the female sex hormones to interfere with that. Probably.
Maybe they even tested and they didn't get the results.
Maybe there was a problem with that. Yes.
Yeah. Well, also because temperature in females might be cycling. No?
That's another point. Yeah. So you need to also get to this right point of the cycle, and if you don't, then you might have a problem to kind of, like exactly. So you need to have it synchronized. Could be.
Okay, go on the last question is, does use of hot drinks in cold weather can help such metabolism? I assume the metabolism refers to the gluconeogenesis. My explanation is it might be affected because the effect of hot and cold drinks on gluconeogenesis is not well studied, but some possible mechanisms existed. For example, hot drinks may increase body temperature, which could stimulate metabolic rate and energy expenditure. This could increase the demand for glucose and activate gluconeogenesis. However, these effects are likely to be small and transient and may depend on other factors, such as the amount and type of drink consumed, the environmental temperature and humidity, and the individual's itself's physiological state and adaptation. And I think it can be studied, but I don't think it's necessary because if you drink hot water for a short time, it may it's difficult to see some changes because human beings can regulate themselves to maintain the homeostasis. But drink the hot water for a long time is not reality because no one can drink water for some longer time. That's the explanation.
Okay. I drink hot water all the time in the form of tea. Okay. Jiang, can you make a short summary? Like, what do you think about this paper, about the authors, or maybe some like what is your final conclusion? Can you make some concluding remarks?
I find this literature because I study about the miRNA, and when I read it, I think they study the miRNA with a provide very solid data. They use knockout, knockdown method, and they restore it. And they also use the CRISPR-Cas9 to generate a knockout mouse model. Unless the dual luciferase method which I used, I still think they are based to study miRNAs. It's great. For other points, for example, the gluconeogenesis, I know very little about this area, so I just read what they wrote.
Okay, thank you. Let's maybe open the discussion. So, like, if you have any other comments or your own conclusions or your thoughts, so everyone is welcome to contribute. Maybe I will start. So first of all, Zixuan, it was a great presentation. You prepared it nicely, and also like this is a very long paper, like a pretty big paper, and you managed to put just 26-minute video, which is concise, very well structured, so really great. So I really enjoyed that. Just a small piece of advice. When you start, can you open the PDF of the paper so that we can have it shared with everyone?
PDF.
Yeah. Do you have it, or not necessary? You can also go here. Yeah, that's fine. Can you go to the figure? Wait a minute. 8.0. The 8 panel O. Yeah. Yeah, the last one. Yeah. So, can you zoom it in? Okay. So, in some papers, they provide graphical abstract. In Cell Press, it's just in the very beginning. Some other journals also provide it in the very beginning. But in Nature, very many journals provide it as the last panel of the last figure, and that is this panel 8.0. And this basically represents the whole story, like the whole concept, the whole hypothesis, and what they found. And it is really great to start with that. So I just, for you, if you're pitching someone and you're trying to tell the story, either you make a poster presentation or you make an oral presentation, talk, or whatever. This could be very useful to first start with the conclusion, because this is the conclusion, the general conclusion, what has been done, what has been found. So you can just start, okay, in this story, they found this, and I will show you later how exactly did they do this. Yeah, so this really helps researchers to structure, to understand what the story will be about, and also it can, some researchers are maybe in the field, they know all of the story, they just don't know this one. They will immediately understand what it is about. They don't even need to know all these details. Some other researchers, it's opposite. They don't understand this topic because they are working on some completely different direction, okay? So in order to understand the context, this is also something very helpful, okay? You have liver tissue there, you have brown adipose tissue, you have this interaction there, and you say, okay, I understand, that's what it is about. It's about this extracellular vesicles somehow signaling to this one, okay? So that is something which you can take in consideration. So this could be really helpful for you to start, and then just go one by one like that. That you made very well. Yeah, you made nice introduction. You put the hypothesis, and also you put the methods. You even structured and compared different methods. And then went towards the results, and then made this discussion and conclusion. So otherwise it's really well done. Yeah. So I still have a problem with this functionality. I mean, I'm quite well convinced about this effect that they secrete. Those vesicles, the vesicles get into the liver, also that there is an increased 378 packaging, and also that it gets to the liver. I'm also convinced about that. What I'm not convinced is how this tiny amount, this really very little amount of 378 can do such a strong effect. Moreover, they show also sometimes that there is, in some conditions where they, for example, make this ectomy, the BAT-ectomy, to remove the brown adipose tissue, and you have decrease of the 378, which is having just very little expression in the liver, because there is basically no expression at all in the liver itself. They claim that all of it comes from the brown adipose tissue. So they see the decline of 378. And while showing this decline, they also show the decline of the output. And microRNAs don't work like that, okay, you increase it a little bit, just 2 times more, and you have the 2 times more increase of the genes which are responsive for gluconeogenesis. You decrease it 2 times, then you see immediately the decrease of those genes. This looks a little bit kind of. I'm not saying that they manipulated, but I'm just saying that the last two figures, they raise questions. This is kind of looks too good to be true. I cannot check the raw data. I cannot see that, but I have doubts. So, dear authors, you will be watching this YouTube later, and I totally understand that this might induce some kind of frustration about like whatever we are discussing here, but this is I would like to address this point of criticism. So I'm not convinced. So before I see the direct interaction of the target with this microRNA within the liver tissue by using, for example, Heat's clip, I just don't understand how can this small amount, this very tiny amount, these microRNAs which are getting from the brown adipose tissue, while the majority of microRNA within the hepatocytes are microRNA 122A and 122B, as well as some other microRNAs, so all the ego should be occupied. How can this work like that? So this is really beautiful, beautifully shown, but I don't fully believe in that. So that is my point of criticism. Sorry for that. Usually when the authors manipulate, as we discussed many times in Journal Club, you can see that. You can see, okay, they are sloppy here, they are sloppy there, there are kind of many typos, they are mixing up, they are making kind of like the graphs which are like not fitting together and so on. They do some p chasing. You can sometimes see these points where they are trying to manipulate. And I cannot say this about this paper. The authors are very accurate. They are not sloppy. I found only one typo. I mean, it is. It doesn't look like they are sloppy. They are really careful. But sorry, those points are still not. So as I said, so like this, like you increase it 2 times from like very little, or decrease it 2 times from very little, and you get exactly the same response for the target genes. 2 times increase, 2 times decrease. It is, and we are talking about the inhibiting the P110 alpha, which is on the very core of the pathway, which is regulated by phosphorylation. So then there is a cascade of phosphorylating reactions. Yeah, GSK3 beta. As well as like FOXO, which is, AKT phosphorylates FOXO in order to inactivate it. So phosphorylated FOXO is inactivated. So FOXO, which is active, will be only if the PI3 kinase is inhibited. The same is for the GSK3 beta. They show it really nicely. But there is a degree of multiplication. So each round coming from this P110 alpha, there is a multiplication of this by the phosphorylation. That's the whole concept of the cascade. So to have exactly like 2 times increase of microRNA, and you have like very bottom, like they claim that P110 is in the very top of the chain, is being inhibited directly, and this causes the change in the genes which are on the very bottom, yeah, so which they measure, like the G6 phosphatase, PEP, and so on and so on, and that this ratio of 2 times is exactly the same if you increase or decrease this microRNA. It sounds suspicious, it sounds too good to be true. I think it is not what you usually see in the biological systems. I am having concerns here. So I don't have any proofs, and I don't have any ways to prove that there is something wrong with this data, because usually we see that. We see those sloppy authors which are trying to hide something or trying to make this p chasing, and we can see these manipulations. Here I cannot detect any kind of problems with the they make this drafting of the figures. But these points, the very last two figures, like all the rest in the beginning, I'm convinced. But if like what they do in the last two figures is not how it is exactly, yeah. So if it is kind of made fit to the hypothesis, then I would of course question the whole the rest of the story. So sorry, authors, again. As I said, I don't have any solid proof, so this is just my concern. So I just raise it, and it will be totally resolved if I at some point see these data. For example, that the ago is packaged together with this microRNA within these vesicles, or there is hit clip data showing that there is a direct interaction of this microRNA with the target within the liver tissue. So hit clip of the liver. If I see this, I will be convinced, and I will be yours. I will tell, I mean, this is a really extraordinary story, and it is an extraordinary story. So I was really surprised when I saw that, but I have my concerns. So I am not fully satisfied, so I'm not fully fine with the evidences which I see. So this is a little bit from my side. So now I let someone else speak. If you have any other points, questions, or notes, concerns. Your point is to cool down only the tissue, right? Anyone would like to address that? Zixuan?
I don't know too much about it.
But you understood the question, right? So Humar was saying that instead of cooling down the animal and giving the shock to the animal, and then studying those vesicles and then trying to use them in vitro, in vivo, and so on, to cool down the tissue per se, so like the brown adipose tissue. Right? Is it the question? Does anybody want to answer that?
I mean, they didn't do it. Like, her question is why they didn't do it, or if it would be useful to do it.
Both.
Because yeah, I mean, it would be definitely interesting, but I don't know if you will have an effect, because since that's not something that would ever happen in real life, I don't know if we'll have the impact enough to activate all these mechanisms. Just a thought.
Okay, maybe I can try. So firstly, in vitro they cultured hepatocytes, and hepatocytes is just an output system. So they tried as an output system where they add these vesicles from the brown adipose tissue. So they don't culture brown adipose tissue. They culture hepatocytes, only the output tissue, not the input tissue. So cooling the hepatocytes doesn't make sense here. Cooling brown adipose tissue would not make sense as well, because there is a mechanism which is driven by the sympathetic nervous system, which drives the increase of the thermogenesis in the brown adipose tissue. So it is all initiated within the hypothalamus. Hypothalamus senses the decrease of the temperature and drives the sympathetic nervous system to increase the thermogenesis. So isolated, so the per se brown adipose tissue is not a thermostat. It doesn't sense the temperature. It just reacts on the sympathetic nervous system. So the sensing module is in the hypothalamus. That is why I think this experiment is kind of like, this is totally adequate system which they used. Okay, so I think the question is, I will try to reformulate if I understand it correctly. You are asking about, like, in the case of a fever context where you have the increased heat dissipation by mitochondria because of the increased activity of UCP1. Whether this per se somehow influences the gluconeogenesis in the liver, and whether this, for example, whether we can expect that there is some influence on the gluconeogenesis because of the maybe changed expression of or packaging of the 378 and then signaling to the liver, right? So that is your question. Zixuan, can you address that?
I don't understand.
I think the question is about the fever. Yeah. So there is increase. And by the way, in that case, it's not only brown adipose tissue, right? So the question is whether we could expect that there is some.
Because I think in fevers, many things, many factors will change.
Yeah, I think it might be it might well be that there is some impact on gluconeogenesis, maybe via the extracellular vesicles. But I think all of this should be studied. You should make somehow the system to remove all other factors which influence and try to study this particular question as these guys did. Yeah, so they focused on only this cold exposed brown adipose tissue effect on the liver, which is a really nice hypothesis, and I think they attempted to nicely show that. So for me, there are some points which are missing. Otherwise, I think they did very well. So similar for that one. So I don't know what to answer. So like without having the data, it is difficult to speculate. It could be, right? Yeah, I think it was pretty interesting. I liked it very much. Like, firstly, Zixuan's presentation and also explanations. I think they were great. Thank you very much for your questions. Really thoughtful. And we were trying to get deeper there. We were trying to assess like what the authors did. I think the authors did very well. If everything is actually true. Sorry for this.
Allegedly.
And I think overall, yeah. So this interaction with two teams, like online and offline, I think it works also pretty well. So we are looking forward to the continuation. So the next journal club will be in 3 weeks, because in 2 weeks we will have a national holiday, so we will need to shift it. But otherwise, it will be every second holiday. Every second Tuesday, exactly. And right. So the next one will be one of the presenters from your group. So you can just choose the paper. You can ask Alex. She can explain about like how to prepare, which software to use, or some recommended software to record the video, and so on and so on. So if you have any questions, any problems, she's the person to ask.
Yeah, all the information is in OneNote, but if you have doubts about it or you want something different, then you can ask me. You can write me by email, or you can write in the chat of the OneNote page.
Yes, thank you, Alex. And thank you, Zhang, and thank you, Fazli, and all the members of your group, Fazli, and also to everyone who is present here. So really inspiring discussion. Thank you very much. Have a great day, everyone. Bye-bye.
Bye-bye.
Thank you. Bye-bye.